I hope you can see me now. I've got a message from YouTube, you're live, so let's hope it works. Sorry about the short delay. We've been trying to work out a few technical issues, but it should work from here on in. My name's Gillian Peterson. Welcome to the next installment of Micro Seminars, brought to you by the Micro Seminars team and now also supported by the ISME Early Career Scientist Committee. I'm the chair of the committee. My name's Gillian Peterson. If you'd like to hear more about us or find out more, just Google ISME microbes and you'll see us there. And feel free to get in touch if you're an early career microbial ecologist and you've got ideas for us. Our speaker today is Flora Vincent. Welcome, Flora, and thank you very much for your willingness to prepare a talk for us, to keep us busy during times of quarantine. To introduce you to Flora, she comes from France. She did a couple of master's degrees before starting out on her PhD. She was first trained as an environmental engineer and then was keen to do a master's program in interdisciplinary life sciences run at the Paris Descartes University. And as part of that training, she was able to do lab rotations. One of those she did with Chris Bowler. And she enjoyed her time there so much that she continued as a PhD student with Chris. And I'm not sure what she'd say about which of the moments during her PhD were real highlights, but it sounds like to me that 10 weeks on board the Tara Ocean schooner during the Tara Pacific cruise was probably one of the real highlights. And something that gave her lots of memories for a time now being locked up inside. For the last few years, Flora has been a postdoctoral fellowship at the Weizmann Institute, funded by a fellowship from the Israeli Academy of Sciences. And her motivation to come on today and present her research is that she's sitting on some exciting new data and realized that the timing wasn't great because many of the conferences she'd planned to go to and present at this year were going to be cancelled. So we're happy to offer this alternative option. And we're really excited to hear about your work on revealing active viral infection in the ocean. So let's switch over to Flora. We're looking forward to your talk.
Thank you. So can you see my screen?
Looks great, Flora. Final message to the audience. Chip in with questions anytime you have a question. Just type it into the YouTube feed and I'll be able to pose these to Flora at the end of the talk.
Okay, so first of all, thanks a lot, Gillian, for that introduction, and thanks to all of you that are watching from a bit probably everywhere in the world behind our computers, and also thanks a lot to the Micro Seminars and ISME for this opportunity. It's actually really cool to still have occasions to share the science with people abroad. And before I get into the hardcore science, I just want to tell you a bit from where I'm speaking. So as many of you, I'm confined also in Israel, where you can see we have those amazing desert landscapes that we unfortunately can't really enjoy right now. And for the past 2.5 years, I've been a postdoc in the lab of Asaf Vardi at the Weizmann Institute, which is an amazing place to work. And as Gillian mentioned in the intro, previous to that, I was indeed quite lucky to do my PhD on the Tara Oceans data, which was a global scale, let's say, research project aiming at describing the marine microbial diversity. And I was specifically focusing on discovering new types of interactions involving phytoplankton. So as you can guess, today we'll be talking about marine microbes, this invisible multitude that comprises over 98% of the ocean's biomass, and most of it is invisible to the naked eye. And plankton has representatives in all the domains of life. And I'm particularly interested in photosynthetic organisms, so cyanobacteria and unicellular eukaryotic algae that are held responsible for over 50% of annual primary production, can fix up to 60 gigatons of carbon annually. They are very good indicators of the ecosystem health. And as you can see on those amazing scanning electron microscopy pictures of diatoms on the left and haptophytes on the right, they come in a huge and crazy diversity of shapes and forms. They're not only, let's say, abundant and diverse, they're also very dynamic. As you can see on this model, this is a model of the MIT from the Darwin project, which models the phytoplankton dynamics in the ocean. And each color here corresponds to a different type of phytoplankton, both prokaryotic and eukaryotic. And you can see it's a highly dynamic succession. both in time and in space, which is constrained by biotic and abiotic parameters such as light, temperature, and nutrients. And in the classic view that we have about the importance of marine phytoplankton, not only do they produce massive amounts of oxygen every year, they are also considered as the basis of the marine food chain. As you can see here, in, say, the classic marine food chain view, phytoplankton will be grazed upon by micrograzers such as copepods, and then this will basically transfer the carbon and the organic matter from phytoplankton up the food chain and support higher carnivores such as fish stocks. And this very classic view of the marine food chain with phytoplankton at the bottom of this pyramid was challenged over 3 decades ago with the discovery of aquatic viruses. As you can see on this picture that was published by Jed Furman in 1999, what they did was to take natural seawater and stain the double-stranded DNA in it, revealing all those numerous small green dots that were interpreted as viruses, both of bacteria and eukaryotic organisms. And this picture kind of really reinforced the growing interest that the field had in aquatic virology and the importance that were given to viruses that were after that estimated to be 10 to the power of 30 in terms of total abundance in the ocean. And the fact that viruses suddenly became such an abundant entity in the ocean really fueled new theories that we had about their role in the marine ecosystem. And one of them is called the viral shunt, which is described here, which basically suggests that upon viral infection, phytoplankton cells will get infected, will lyse, and the organic matter will then be made available under the form of particulate and dissolved organic matter that will be recycled by bacteria instead of being made available for grazers. So there is really this idea that the viral shunt will basically transfer the carbon, and instead of going up the food chain, will basically go down to the microbiodiversity. So there is really this idea that those small marine microbial interactions will really impact both the microbial community, but also the nutrient cycling. And despite this importance, we also know since the past 30 years or 20 years that among those numerous small green dots, we have free-floating genomic DNA. We have vesicles that are small cargoes that can also contain DNA and that are not viruses. And most importantly, that a lot of those small viruses could potentially be inactivated viral particles, so not infectious. Which brings us to think that more than really knowing how many of those viruses are in the ocean, the importance is to know how many of them are actually infectious. And this brings me to the viral cell concept that was suggested by Patrick Forter a few years ago, that suggests that the viral cell actually corresponds to the living form of the virus, whereas virions, those small green dots, are in fact the equivalent of seeds or spores, and that we're kind of wrong when we claim that viruses are 10 times more abundant than cells in most environments, because in the end, the important point is how many of those viruses are actively infecting a cell. And furthermore, a few years earlier, in this review by Curtis Suttle in Nature, he really stated the fact that accurate estimates of virus-induced mortality were still elusive, and that we didn't really have proper ways to actually estimate the proportion of cells that died from lysis. And so this is the major bottlenecks that we identified, at least for us in aquatic virology, the fact that we don't know what is the fraction of virus-mediated mortality in the ocean. We don't know how to link subcellular and population dynamics processes of phytoplankton viral infection. We don't really know how this viral infection quantitatively affects the rate and fate of biomass aggregation and sinking, and whether or not this viral infection is actually synchronized in some kind of way with other agents of mortality. And what we believe is that if we manage to start answering some of these questions, we're really going to reach a better understanding of the role of viruses in marine ecosystem in terms of evolution, biogeochemical impact, but also modeling. And what we identified is that there's a need for scalable and mechanistic approaches to characterize this highly dynamic process of microbial interactions. So in order to address those questions, in the Vardi lab, we work on a eukaryotic unicellular algae called Emiliana Huxley. As you can see on those pictures, Emiliana is a small cell of about 5 micron that produces those beautiful coccoliths made of calcium carbonate. This is why it's held responsible for 1/3 of ocean calcification. And every year, Emiliana Huxley can form those massive blooms that you can see from space, due to the light scattering by the coccoliths, that over geological timescales through sedimentation will form those beautiful structures such as the White Cliffs of Dover's. And it is believed that those massive blooms are terminated through biotic interactions that involves a virus, which is called EHV for Emiliana Huxley virus. EHV is a large double-stranded DNA phycodnaviridae that has around 500 genes, some of them encoding pathways for glycosphingolipid synthesis. And this is the virus that's held responsible for bloom demise, even though we don't really have quantitative estimates for that. It's around 200 nanometers large, and it's thought to have a burst size of around 1,000 virions, meaning for every virus in, we have 1,000 viruses out. And actually, one of the first questions that we wanted to answer just by looking at this satellite picture is basically what's the fraction of infected cells in a phytoplankton bloom? And in order to answer that question, we developed an approach that we called virocells. And the idea of virocells is to visualize active viral infection through the co-expression of host and virus messenger RNA. We start by harvesting algae, both in the lab or in the natural environment, and we stain the single cells with fish probes, fluorescent in situ hybridization markers. And those probes basically target messenger RNA, both of the host and of the virus. On the host side, we target PSBA. PSBA is a chloroplast encoded gene that encodes for the D1 protein of photosystem 2. And basically, expression of the PSBA is a good proxy for the host metabolic status. If you're transcribing PSBA, it means that your photosynthetic machinery is active, and it suggests that you're in a good metabolic status. And on the viral side, we target two different genes. One of them is an immediate early gene. It's a viral gene that is expressed at the very beginning of the infection. It's a hypothetical protein. And we also target MCP, which is major capsid protein. It's a structural protein important in the steps of virion assembly, and is expressed more towards of the late stage of infection. And to give you a bit more, let's say, technical details of how those probes work, let's say that we target the PSBA messenger RNA. We'll start by synthesizing small probes of around 20 nucleotides long. Each of those individual probes will be conjugated to a fluorophore that has a specific excitation and emission spectra, so much so that in the end, we have a pool of probes that are specific to the PSBA gene, and that the signal intensity in that specific fluorophore will be proportional to the abundance of your messenger RNA. So in the end, we get single cells in which single molecules of messenger RNA are individually probed. And we really talk about active viral infection because we're targeting virus messenger RNA, and you can only have viral messenger RNA when you have active infection. So once this is done, we move to quantitative visualization, either through epifluorescence microscopy or imaging flow cytometry. So what does it look like? We did a small experiment where we just took samples of 8 and 24 hours post-infection. So basically taking a Emiliana Huxley culture and infecting it with the EHV virus that we have in the lab. What you can see here is a picture in the DAPI channel. You can see here around 700 or 800 cells. You can zoom in and see the small nuclei. This is the picture 8 hours post-infection, and 24 hours post-infection. We see also a few cells, actually many cells that are still containing DNA. On the PSBA side, this is a host messenger RNA. You see that 8 hours post-infection, most of the cells are red, meaning that they are actively transcribing the host messenger RNA, so they are metabolically active. But on the other hand, 24 hours post-infection, there's a complete shutdown of the photosynthetic transcription, and no cells are expressing PSBA anymore. On the viral side, you see that 8 hours post-infection, a few cells are bright yellow, meaning that they are actively transcribing the messenger RNA of the virus. And on the other hand, 24 hours post-infection, you see that most of the cells are bright yellow, meaning that they are really all actively being infected. So we can go a step further and see that at the single cell level, this is a unique single cell. You can see small purple or pink little dots. These are individual molecules of the major capsid protein RNA. So this is really a single molecule resolution level, even though in our case, we're not really interested in single molecules, but rather on a yes-no question of expression of the mRNA in large amounts. So this is in epifluorescence microscopy, where we basically take our preparation, put it on a slide. It's amazing for lowly expressed gene. It's very high resolution, but the problem is that it's a bit low throughput, quite variable in terms of result, and very suboptimal in terms of compensation. This is why I decided to move to imaging flow cytometry, where the idea is that this time the preparation goes to a flow cytometer, where each cell are ordered in a stream, will be excited by lasers with different wavelengths. And this is the picture that you get for each individual cell, whereby one cell has each single cell, has a picture in the bright field, has a picture in the viral messenger RNA channel, in the DAPI and in the host. So much so that you can see that this cell, for example, the second one, does not express viral messenger RNA, but expresses the host messenger RNA, that this cell here expresses the viral messenger RNA, and that this cell here seems to express none of both. So this is interesting because suddenly it becomes quantitative, meaning that we can also perform and conduct time series. This, for example, is a time series from 0 to 56 hours post-infection, where you can see that at the beginning of the infection, most of the population is below this dotted threshold in terms of viral messenger RNA. And suddenly, 4 hours post-infection, you see the rise of this small subpopulation that seems to have high levels of viral mRNA intensity, meaning that this is your population of cells that are actively being infected. On the host side, it's a similar picture. You can see that 0 hours post-infection at the beginning, most of the cells are actively transcribing PSBA, the host photosynthetic gene. And throughout time, there's a gradual fraction of the population that starts to shut down its photosynthetic machinery and move below this positive threshold. And the first thing we wanted to do was basically to apply this assay to the most basic experiment ever, and to really try to connect the subcellular scale with the population dynamics. So what we do is that we basically take a culture of Emiliana Huxley and infected it with EHB-201, which is a virus that has quite high virulence. And it was infected to a MOI of 5 to guarantee that at time point 0, every cell encounters an infectious particle. We follow the cell abundance using analytical flow cytometry. You can see here that the infected culture starts to demise after 48 hours when the control still grows correctly. And you can see here on the black curve, the fraction of cells that are considered to be dead based on cytox positive stain, which mimics quite well the cell abundance. And this is the picture that you see when we look inside on the host expression. What you can see here in red is the fraction of cells that are actively transcribing the photosynthetic gene. In the control culture, 100% of the cells are always expressing that gene. Whereas in the infected culture, between 8 and 24 hours, there's a complete shutdown of the photosynthetic machinery. And overnight, all of the cells completely stop to express the PSBA gene. On the viral side, what you see here in purple is the accumulation of extracellular viral particles. So what is emitted in the media, and you can see that the first increase occurs between 4 and 8 hours post-infection. And then when we look at what's happening inside, this is the fraction of cells that are actively expressing the messenger RNA of the virus. And you can see that 4 hours post-infection, we already have above 15% of the cells that are infected. We're reaching a maximum of 75% of the population being actively infected 24 hours post-infection, and then it follows a gradual decrease. It becomes interesting when we also overlay the percentage of cell death in this program, because you rapidly see that based just on cell counts and cell deaths and maybe viral particles, you don't see much change within the first 24 hours. However, if you look what's happening inside the single cells, you can see that within 24 hours, there's a drastic change in the metabolic status of the cell. And by comparing those three curves together, we started seeing very interesting things, notably what you see between 4 and 24 hours, which is a huge amount of viral release, but very little cell death. When you look at the black curve, you see that very little cells die, meaning that there are not a lot of cells that die, but a lot of cells, but a lot of viruses that are emitted, which really led us to think that there was another mechanism of viral release that was not only through lysis compared to most of viral infection in phytoplankton, and that maybe there's another mechanism that induces the early release of viruses by cells that are not dead yet. And this was actually also suggested in a paper from 2009, where they show small evidence of budding in specific Emiliana Huxley cells upon infection. So this was really starting to question the virus life cycle. The second interesting point that we saw is what happens 48 hours post-infection. As you can see here, in the black curve, you see that there's a huge amount of cells that die, and yet there is no viral accumulation. So we have a large amount of the culture that is crashing, but absolutely no viruses that are released, which led us to question another point about the virus life cycle in this case, which is maybe that those cells are not dying from viral lysis. And this is what we tried to investigate to see if we can detect the cells, that escape viral lysis. This title is really bad, sorry. So before trying to estimate cells that die from viral lysis or not, I just want to spend a bit of time explaining how we define population gates in imaging flow cytometry. So what you see here is a sample taken 0 hours post-infection. Each cell here, each dot here, sorry, represents a cell. You can see that most of the population is in this bottom right corner. The x-axis represents the host messenger RNA, which is basically a proxy, which is basically through the intensity of the messenger RNA of the PSBA probe. And this axis, the x-axis, defines whether you're metabolically active, so PSBA plus, or metabolically inactive, PSBA minus, because we consider that photosynthetic activity is a good proxy for your metabolic status. And the y-axis here is basically the virus messenger RNA. It's the intensity of the MCP messenger RNA probe, and this defines whether you are infected, MCP plus, or non-infected, MCP minus. And by combining those two axes, we basically define 4 subpopulations that are here, that are a combination of the MCP and the PSBA expression. So what you see 0 and 1 hour post-infection is that most of the cells are maintained in this bottom right gate, meaning that they are expressing the host messenger RNA, but no virus messenger RNA. And by the way, all the control cells stay in this gate for most of the experiment. And this is what they look like under the image stream. And 4 to 8 hours post-infection, we have the appearance of this very interesting population here in the top right gate, which is both metabolically active and actively infected, so expressing the MCP messenger RNA. And this is what it looks like. You can see that it has signal in both the virus and the host channel. And 4 to 32 hours post-infection, we have 2 new subpopulations that appear. The first one is the yellow one here, which are basically cells that were actively being infected and that shut down the photosynthetic machinery, so no traces of host messenger RNA anymore. And this very interesting population here on the bottom left corner that are the gray gate, which are basically cells that are not infected, or at least not expressing the viral messenger RNA, but that are gradually losing transcription of the host metabolic gene. So we can actually quantify the relative abundance of those different populations through time and see the succession of those 4 gates. And what we think is happening in this gray gate is that those cells that have a very specific transcriptional signature can correspond to different cell fates. Some of those cells can be dying from programmed cell death induced by neighboring signal. Some of those cells can be in a kind of dormant stage, so a potential pool for resistance. And last but not least, some of them cells can also be infected, but probably in a process of abortive infection because they manage to halt the infection before it reaches the late stage of the major capsid protein expression. And when I see this picture 24 hours post-infection, it really makes me think about when we do bulk studies, that we will filter our cultures and do bulk RNA-seq and basically see the relative upregulation or downregulation of specific genes. And I'm trying to wonder how it will be affected by the fact that we have a very strongly mixed population of cells that have very different transcriptional states. So the next question, because now we could identify those cells that are probably not dying from viral lysis, was to see if we could identify the population that was responsible for early viral release. And to do so, we need to zoom in into what's happening at the beginning of the infection. This is what you see here. In black, at the background is the fraction of cells that are dying, so basically none. And in blue is the accumulation of viral particles. So again, between 4 to 8 hours, we have strong accumulation of viruses in the media. And this is the fraction of cells that are in the 2 infected gates, metabolically active or non-active. And what we decided to do was to compare the DNA content of those 2 populations through time. This is what you see here, 4, 8, and 24 hours post-infection. And this is the DAPI intensity. And what you can see, for example, 4 and 8 hours post-infection is that the cells in this bottom right gate have much higher DAPI content and significantly higher DAPI content than the non-infected cells that you can see here. And in a sense, all the viruses that are present 8 hours post-infection can only be a product of what happened a priori in the past time points. So what we suggest is that those cells are actually completely responsible for early viral release. The reason they have much higher DAPI intensity is because they probably induced de novo nucleotide synthesis to meet the high demands of a giant virus that has over 500 genes in the genome. So this explains that, this shows at least that host nucleotide recycling is not enough to meet the high demands of this early viral release, and that the induction of de novo nucleotide synthesis is necessary to create this early chronic release. So we basically reach a kind of a model at this stage where at the beginning of the infection, cells are metabolically active and non-infected. And upon presentation to the virus, there are basically two major routes. The first one is that they will get infected. The metabolically active cells, so that are still photosynthetically active, will basically bud viruses. And upon some kind of signaling or switch in the viral program, they will shut down the photosynthetic machinery. And those cells that are metabolically inactive and infected will probably lead to viral lysis. On the other hand, the second pathway is that upon infection, cells will not undergo the viral program to its end, but instead have metabolic inactivation, so stop transcribing messenger RNA in the photosynthetic machinery. And those cells most likely might undergo programmed cell death due to signaling from neighboring cells that are infected, could be a pull for resistance, but could also die from abortive infection. So of course, once we had that in the lab, the second, let's say the natural route was, okay, let's go to the real stuff. What happens in a natural bloom? And for this, we decided to go a bit wild, and we were very lucky to be able to conduct a mesocosm experiment in Bergen in May 2018. Bergen is in Norway here, and the facility is amazing to study E. Huxley natural blooms. And the setup is the following. We had 7 different bags of 11,000 liters each, and the bags were filled with natural water from the fjord. So we really start with a natural inoculum of a natural population that we know is supposed to contain Emiliana Huxley. And we supplemented those bags, those impermeable bags, with nutrients, nitrate and phosphate, and basically crossed fingers to have a phytoplankton bloom that would ultimately lead to E. Huxley blooming in the field. And it was an amazing consortium with people from everywhere in the world that worked very hard for a month to conduct this experiment. So first thing, we were very lucky because we had a bloom. So this is what the bag looks like at day 0. You can see that the water is quite translucent. And on day 21, the water turns completely milky, and this is due to the coccoliths, the calcium carbonate coccoliths from Emiliana Huxley. And you can see on the scanning electron microscopy, it's not very ambiguous. These are complete E. Huxley cells that were filling our bags. So we followed the dynamics of Emiliana Huxley blooms in the 7 different bags. This is abundance of Emiliana Huxley cells through time, monitored by flow cytometry. And you can see just based on the trend that we can define 3 major phases in this bloom: the pre-bloom here, the bloom exponential growth of our algae of interest, and the last phase, which is the demise phase. Each line corresponds to a different bag. And I'm going to focus particularly on bag 4 here in red, and on 2 periods that are the bloom and the demise. And the goal is really to try and track active viral infection during those times. So the problem when you go wild is that it's cool, but it's also complex. And as you can see on this eukaryotic tree of life, marine microorganisms basically populate the whole tree of life. So we really had this challenge of being able to specifically identify Emiliana Huxley, because contrary to the lab, we're not working on an isogenic cell line again, or culture, sorry. And we really had this challenge of identifying Emiliana Huxley. And to do so, I built upon work from Miguel Frada and designed a probe that targets specifically the 28S ribosomal RNA from Emiliana Huxley that are incorporated in the viral sailfish. So much so that at the end of the day, we can get for each single cell a picture of the 28S, which enables us to identify specifically Emiliana Huxley. Also a picture for the viral messenger RNA, for the host messenger RNA, for the DAPI, and additionally for site scattering, which is a proxy for calcification, which is important in our case. So what do we see? So these are times, sorry, I'm missing a legend here. This is the abundance of Emiliana Huxley through different days. So each number here is a day. It's separated by 24 hours. This is the abundance. You can see that we reached quite high numbers of over 80,000 cells per ml in bag number 4. We also tracked the abundance of viral-like particles, as you can see here in purple, that start accumulating on day 17. So this is also monitored by flow cytometry. These are viral particles per ml. And then when we conduct the single molecule fish, the viral cell fish, we reach, you can see these are the orange bars here that are basically the fraction of Emiliana Huxley cells that are infected through time. And you can see the scale here. These are the fraction of MCP plus cells. So there are several things to get from this graph. First is the first assessment of active viral infection in natural population. So we're quite happy about that. And then what you can see is what happens on day 17. So day 17 here is the peak of the bloom, 80,000 cells per ml. And we barely have less than, let's say, 3% of the cells that are actively being infected. And overnight, within 24 hours, you basically lose 50% of the cells, moving from 80,000 to maybe 50,000 cells per ml. And in terms of infected cells per ml, we have around 20% of the cells that are actively being infected. So what happens in those 24 hours? We have several options. One of them that is quite pessimistic is that we might be missing an intermediate time point here where we would have a very high fraction of cells that are infected that would explain that we lose so many cells overnight. And the second option is that we just don't go beyond 20% of infected cells throughout an Emiliana Huxley bloom, and that somehow cells in this phase overnight kind of died from something else and then viral infection took over. And if you work within this assumption, it means that we never have above 27% of the cells are infected during the course of a bloom. So there are several things that we wanted to test from the lab. For example, we compared the DAPI intensity between actively infected and non-infected cells. You can see here through time that the infected cells constantly have much higher DAPI intensity than non-infected cells, which kind of supported the idea of de novo nucleotide synthesis in actively infected cells. We were also able to quantify the relative abundance of the four populations through time, and you can see the succession of cells that are in green, metabolically active and not infected, that are quickly replaced by the yellow cells here, that are already cells that shut down the photosynthetic machinery but are actively being infected. And moreover, the very high proportion of cells that are double negatives, meaning that they are cells that will not lyse from viral infection. And just to touch upon that, that point, one of the things that I think is happening also is that between day 17 and day 18, what we are actually missing is this transition by the double positive state. That we see cells on day 17 that are starting to be infected, and probably in the course of those 24 hours, they are moving up to be metabolically active and infected. They are releasing viruses, and overnight they are switching off the photosynthetic machinery, because we know from previous really interesting work from Kim Sramatakon that light is really a big player in the viral infection, and they probably turn off the photosynthetic machinery overnight, and this is what we see on day 18. So probably we're missing kind of an evening time point here, but not sure that we're going to go beyond 30% of infected cells anyhow. So those results on this bag, we also performed a single cell RNA-seq using 10x technologies. Oops. This is, in collaboration with Gurf Khevroni, a postdoc in my lab. And this also confirms this very small number of around 25% of infected cells, which bring us to the natural question of, if we only have 25% of infected cells in a media, what becomes with the rest? And this is when we start to try and zoom out from the virocentric point of view. Here, what you can see is the host abundance, so the Emiliana Huxley abundance through time. And this is the virus abundance. And you can see that, even though we have some viruses in most of the bags, it's really not in equal amounts. And especially the most abundant host bags don't necessarily have the highest amount of viruses. So it means that despite the fact that all bags crash, it's really not with an equal amount of viruses. And when you think about it, it makes sense. Emiliana Huxley can, of course, die from viral lysis, but it can also die from pathogenic bacteria. This is work from Noah Barak in my lab, but also through allelopathy, cell-cell communication. We know that infected cells secrete molecules that can induce programmed cell death in neighboring cells, but also grazing from micrograzers. So this is what we're currently also trying to interpret in those different bags, to try and see what are the different agents of mortality, and if we can actually quantify them and their impact on the microbial succession and nutrient cycling. So anyhow, whatever the form of demise, whatever the cause, the biomass still needs to go somewhere. And this brings me to whether or not and how viral infection can affect the fate of biomass in the ocean. And for this, we need to touch upon what happens when single cells become aggregates and go back to theories that were previously developed of the shunt and shuttle paradigm. Basically, the viral shunt that was developed by Suttle in the early days suggests that upon infection, marine microalgae will basically lyse and the organic matter will be kept small under the form of dissolved and particulate organic matter, and that this will basically reduce the food supply to larger eukaryotes, including fish stock, carbon flux, etc. And a few years later, there's a new hypothesis that emerged, which is that of the viral shuttle. And the idea of the viral shuttle is that upon infection, and it's not an idea, it's a fact, upon infection, phytoplankton will produce transparent exopolysaccharides that will induce aggregation. So those sticky viral lysets will lead to aggregation that will ultimately induce sinking and increase the carbon flux to the deep ocean. So whether or not we're in a shunt or shuttle paradigm will really affect how the marine microbial community will respond, and especially how the carbon will basically be transferred in the whole ecosystem. So what we wanted to see was whether or not we could detect and measure aggregation. And one of the objects, one of the main points of the imaging flow cytometer is that you can actually see your objects. And very rapidly, I started seeing those phenotypes that are kind of blobs and objects that are around 10 micron large, that have DAPI signal, that have PSBA signal, that even have MCP signal, meaning that what you're seeing is basically an aggregate containing cells that are either metabolically active or infected. And by digging a bit more, we realized that we could actually differentiate aggregates from single cell based on the signature of the DAPI area versus the DAPI circularity. We can identify this big population here of single cells. This is on the population on which I base most of my analysis. Doublets here that are also present, that are most likely an artifact of the processing. And this last population here that are aggregates, that are objects that have an area of above 60 micrometers. And then the good thing is that once you have a population, you can basically quantify it. And what you see here is the fraction of all those elements, the fraction of this total population that is under the form of aggregates, with clear accumulation of those large objects in infected cultures compared to non-infected cultures. And most importantly, we can characterize those aggregates. We can basically detect whether or not they contain actively infected cells and whether or not they contain metabolically active cells. And if I were just to draw you the bottom line from the graph here and the graph here, at the beginning of the infection, aggregates are not numerous. They are scarce, but most of them are highly infected and highly active in terms of PSBA transcription. And towards the end of the infection, aggregates become much more numerous, but they do not contain infected cells and do not contain metabolically active cells. And what is interesting, and actually what the fate of those aggregates at the beginning or at the end will really affect what type of microbes will be attracted to it and what type of reservoir of viruses we are creating through time. And one important point also is what we see here 8 hours post-infection, that basically 75% of those aggregates are infected, which is quite higher than what we see in the single cell data, where in the single cell data, we only have 50% of the population that is infected, which means that there is an enrichment of infected cells in those aggregates that are most likely going to sink faster. And for us, even though it's complete hand waving, we kind of foresee this as a good host defense strategy, whereby upon infection, you're going to enrich the aggregation of infected cells so they will sink and get out of the pool of non-infected naive cells and avoid further propagation of the virus. So the fact that we have more aggregation towards the end of the infection and very little at the beginning kind of gives a feel that maybe we have a bit more shunt at the beginning and maybe a bit more shuttle at the end. And of course, it wasn't going to be a binary answer, and it's something a bit more gradual, but clear identification of accumulation of aggregates through time and characterizing their nature. So we're also quite happy to be able to identify it in the field. We can actually see, for example, aggregates that contain, Emiliana Huxley cells that are infected. However, the quantification is a bit more tricky, and we're working on it to quantify the accumulation of aggregates in natural samples. So now I just want to kind of wrap it up by showing you a bit what we're working on right now. So kind of what's next. Here, what you see is a picture that is quite binary, because I'm only looking at a yes/no answer in terms of metabolic activity and viral infection. But in fact, the image stream gives you for each individual cell over 700 features that describe its morphology and all types of metrics. So it's a lot of data. And right now we're trying to move to something a bit more continuous, less binary, to try and really reconstruct the trajectory of a single cell when it starts to be infected and when it reaches two different or different fates or morphological features. So for this, we're working with Maxime Lippeveld from Leuven to try and build a more continuous, let's say, trajectory of cells upon infection. And the idea is to actually, in the end, combine it with single-cell RNA-seq data that we have in the lab. Amazing work from Chuan Ku and Uri Shane, where they performed also time course of single-cell dual RNA-seq using MARSEQ technologies. And the idea here is between the virocells that provides the morphology and the single-cell RNA-seq that provides the transcription, we can actually try and combine them together and really build the morphological and transcriptional trajectory that the single cell undergoes upon infection. Among the other lines of research that we're trying to work on now is to try and revisit viral dynamics under the subcellular lens, and basically applying viral sailfish to tons of different conditions, because it's quite now implemented assay in the lab. We have, we would like to try it with different viral host strains, with different light and temperature regimes, with different nutrient starvation. So basically trying to disentangle the effect of both abiotic and abiotic factors on viral dynamics, different cell densities, but of course also different genes and more field samples. So this is work that was started also by Elad Yitzhaki, which is a student in our lab. He started applying viral sailfish to different viral pressures. So how does the viral load, the multiplicity of infection, affect the dynamics of the host and the virus? And the bottom line is basically that at low MOI, at very low MOI, it seems that we have more signaling for non-viral lysis, which also supports what we see in the lab. And this is what we're currently investigating. One of the other avenues of research is based on this picture, which basically shows a few cells here. You can see a few dozens of cells. One of them is circled in white here. You can see the DAPI signal. You can also see that it's expressing the host messenger RNA. It is not expressing any MCP messenger RNA, but what you can see here is that it has a very strong signal in the immediate early gene. So one of the viral genes that is expressed very early on in the viral program. And when you compare the intensity of the signal through this white line here, you can rapidly see that the red signal, which is basically the PSBA, the host messenger RNA, overlaps and is colocalized with the immediate early gene. And by doing stats on a few hundred cells, we basically see that statistically speaking, the immediate early genes tend to be more colocalized in the chloroplast than in the perinuclear region. What is it doing? We don't know. How is it getting there? We don't know. But we find it really exciting because it's really starting to look at the level of organelles and really how the virus is hijacking the photosynthetic machinery of the host. And this also rings bell with cyanophages and PSBA, even though mechanisms are probably not the same. It's really interesting to see that between photosynthetic hosts and viruses, there is something going on definitely at that level. So what are the take-home messages? Basically, we're addressing quite an old question with new approaches, providing the first quantification of virus-mediated mortality in natural phytoplankton. We also provide a new way to quantify aggregates and to connect really the subcellular dynamics with the population level tendencies. And beyond being a cool tool, it also really raised questions about giant virus infection cycles in the ocean, confirming the idea that we had metabolically active cells that were responsible for early viral release, that we had different cell fates that involved also the intervention of other mortality agents, metabolic hijacking, confirming really the idea that a subpopulation of cells will undergo de novo nucleotide synthesis to meet the high demands of the virus, but also how defining the aggregate composition can hint towards specific host defense strategy. So for us, we basically have this new tool that combines morphology, host virus gene expression, it's quantitative through time, and it can actually be deployed to very diverse microbial interactions, both in the lab and in the field throughout the various scales. So we hope that kind of 3 decades later, we we kind of complement that picture with a new picture showing actually those active viral cells that would be the small dispersed yellow dots. So with that, most importantly, thanks to Asaf Vardi, who's really an amazing PI to work with. Uri Shane, a former student in the lab that's been really instrumental in kickstarting this project a bit more than a year and a half ago. Ziv Porat, and that trained me on everything that I showed you today. Ziv Porat from the imaging flow cytometer, that's also been amazing to work with. And last but not least, the whole Vardi lab and our departmental staff that I really miss right now. And thank you for listening everywhere where you are.
Okay. Thank you, Flora. That was an excellent talk, very enjoyable. Do you want to end the screen sharing and we can switch back? So I wanted to pick up on your last, or one of the final points. You talked again about the nucleotide synthesis and how important this must be during the early stages of infection to produce new viral particles. And is there anything that you see in the single cell RNA-seq data that shows in the host that this really is expressed at a higher level?
So actually, I don't want to say mistake. I don't think we see it on the host side in the single cell RNA-seq data yet, but need to confirm with people in the lab. I know we see it in bulk. For sure, we see in bulk induction of nucleotide synthesis pathways. It's not only work from Shiloh Rosenwasser in our lab, but also a recent paper from Kim Samatrakom that really shows induction of pentose phosphate pathways that are important for de novo nucleotide synthesis. So we do definitely see it on bulk. In the single cell RNA-seq, I'm less sure about it. So this is part of the ongoing projects to really try and pinpoint specific questions and combine them together. Yeah, definitely.
Yeah, it's really amazing to break it down to the single cell level. You're really starting to question some of the assumptions on why these blooms crash, for example, that viral lysis is the major mechanism. That's really cool. We have a question from Erin Eggleston. So firstly, thanks for an excellent talk. And do you have ideas for applying virocell fish to multi-species blooms with various viral players? I imagine the limitation is the FC channel number or a priori probes.
Yeah, that's a great question. To be honest, I think one of the recent, I think it's more a question of methodologies. Now, in our case, for sure, the fact that we are limited by the different channels limits the amount of diversity that we can target. But I know of work now that is based on, all this HCR technology, more based on microscopy for the moment, epifluorescence microscopy, not flow cytometry, where you can really filter your sample, fix it on a slide, and basically stain with specific probes, take a picture, wash it away, stain again with new probes, take a picture, wash it away, and basically you're multiplexing, and this is how you can add new species. So I know this is starting to be possible, not yet completely applied to natural samples, but I definitely think this would be the next step. And I also think it's different methods for different questions. One of the major advantages of the viral fish is that it's extremely quantitative, because for each sample, you're acquiring over 80,000 cells. So in terms of statistics, you're very robust. But then for applying it to microbial, really complex communities where you want to target several hosts and several viruses, I think it will take a few more years before we get there. But HCR is probably a good way to get there.
Great, thanks. We have another question, from somebody with a cryptic name who starts off, awesome talk, thanks. And how or what role do you think extracellular vesicles might play in these dynamics?
Yeah, so we have Daniela Schatz in our lab, who's really, this is exactly her topic of research, where she discovered that vesicles indeed played a huge role during viral infection. Both by extending the half-life of the viruses, so potentially vesicles have a role in let's say, maintaining the half-life, extending the half-life of the viruses, but also as a communication cargo. Like, we don't really know exactly what's happening. I do believe that they are major players in the cell-cell communication also, because infected cells do release tons of vesicles, and probably a good cargo for chemical signaling that can also induce some kind of maybe cell death in neighboring cells or more resistance. So yeah, definitely major role of vesicles, I would say, as a cargo for signaling.
We also have a question from Gal Ophir. Clap, clap, clap. Can you quantify how much of the final viral yield, so I guess the total viral yield, is released pre-lysis?
By chronic release, basically.
Mm-hmm. Like the early stage release that you were talking about, I think that's
yeah. So between so hi Gal, and yes we can, and I didn't do it. Good reminder that I need to do it. But yeah, technically, basically, I think the most interesting time point is between 4 and 8 hours post-infection, which is really the moment where you see viral release, and we know exactly how many cells are metabolically active and infected, and we can see that there's no cell there. So this is exactly what we need to do to quantify. And now what we're starting to do is also to play with the different viral loads. This is the work from Elad in our lab, which basically tells you that this number changes. It will change depending on how much viruses you put at the beginning. It will change on the cell density. The number of viruses that will be emitted by a single cell through chronic release is not a fixed number. And this is where it gets interesting, is that we can really quantify how it changes based on different parameters of a host virus dynamics.
Cool. We also have a question from Maya Breitbart, who has asked us, do you know what the signals and mechanisms of the abortive infections, or PCD strategy, are? With that many cells in a bloom, is this essentially self-sacrifice for the greater good? Very interesting question.
Yeah, great. So in terms of abortive infection, I don't say I'm sure, but we don't know much about the abortive infection in EHUX, unfortunately. But great avenue for next projects. In terms of PCD, we can say that infected cells, this is work from Asaf 2012, that infected cells produce viral glycosphingolipids that will induce cell death in neighboring cells. Exact mechanism, we don't know. And in terms of greater good, yes, it's really possible that in the end, one thing that we have to keep in mind also is that in the lab, we're working on the isogenic strains. So a single strain of Emiliana Huxley. In the field, it's most likely that we have several strains. So this, I think, is another component that we need to keep in mind. When you see the bloom and crash of Emiliana Huxley in the field, I don't believe we're looking just at a single strain that is blooming and crashing. There's probably a succession of those strains in there. So when cells are dying, are we really looking at all the population that is crashing, or is it only a specific strain? Probably the strain that will die from programmed cell death and avoid further propagation of viruses is the one that is susceptible, obviously. But yeah, could be. We need to test.
Cool. And just because I'm curious, I'm going to ask a final question, and that's about as a technical question about the detection limit of the virocells method. I mean, and you mentioned that you had the single cell RNA-seq to back that up, and you mentioned that you thought it was quite a sensitive method, but do you have maybe some classical methods such as RT-qPCR or something that would show you what is the detection limit?
Yeah, very good question. So no, we didn't do RT-qPCR for the moment. In terms of epifluorescence microscopy, my detection limit is technically 1 molecule. I can see 1 molecule of messenger RNA. In the imaging flow cytometer, it's a bit of a different story. The resolution doesn't enable us to detect a single molecule. What I think our detection limit is that we will start seeing a signal in the imaging flow cytometer after, let's say, 100 molecules of messenger RNA. So once I have 100 molecules, it's a bit back of the envelope, but from a few calculations, this is what we think we see. And we combine it with the single cell RNA-seq data, which also has its own limitations, right? In single cell RNA-seq, you only capture 5% of the transcriptome. So by combining both together, checking up the dynamics, I think, we start seeing cells when they have 100 molecules of messenger RNA. And in our case, at least until now, we're not so obsessed by that detection limit because we're really interested in cells that are actively infected. And when you're producing at least 1,000 virions, you don't care if you have 1,000 or 2,000 molecules or 10, like just 1 molecules of messenger RNA. Considering that for a single virion, you need maybe 1,000 major capsid proteins, you will need at least 1,000, like not 1,000 RNA, but anyway. So I would say 100 messenger RNA. That's our detection limit.
Yeah, true. Interesting point. Now, I'm just fascinated by these numbers from the field experiment that you've got, maybe 25% infected cells, and that's the most you ever see. I think that's a really interesting result.
Yeah, I think so too.
Okay, well, thank you, Flora, and thank you everybody for joining in today, and thank you for sticking with us. I saw that most people, you all stayed with us, even though in the beginning we had a slight delay. So yeah, it was worth staying on for. Thank you very much, Flora.
Thank you very much.
Lots more. You're welcome. We have lots more micro seminars planned, so check us out on the Micro Seminars website and join us again. I think the next upcoming seminar will be next week. And we're going to continue these seminars. The feedback has been great. So if you're still interested in giving a seminar, maybe you've got some new interesting data, also feel free to get in touch or send an application on the ISME Early Career website that's set up for that. Okay, stay safe, everyone. Stay home. See you again soon.